So, I don't know, Madam anataka kunialika kama niko tu sawa. I can just proceed. Sawa sawa. So once again, good morning. Good morning. Because I'm nasikia. Good morning.
Good morning.
So we shall, I don't know, we shall take a little bit some time. We can take even 1 hour or 2 hours. Or even you can take the whole day to discuss the whole thing about this biology. But one thing I want us to look at, I just want us to look at briefly, we shall not go into details, okay? We shall look briefly about what you are supposed to do. I know you are ready, you are prepared, you have been reading for the last 4 years, so you are ready. So what I'm going to do is very simple. Just show you some of the tips that maybe you require. Not even necessarily tips. These are just some of the reminders that we're supposed to tell you. The key areas that you are supposed to make sure that you give them a maximum attention. Attention is what is important. So first of all, this is biology. And biology has 3 papers. Yes. It has 3 papers. The first paper is biology paper 1. Biology paper 1. Biology paper 1 is 80 marks. 80 marks. So out of 80 marks, biology paper 1, what nasema, it is what we call short answer questions. Short answer questions. And that's why you discover that it has between 25 to around 30 questions. So if you take an approximation, if you have 25 and above questions, that means, and our target is 80 marks, that means it's very rare for you to get a question carrying 4 marks. So we find that for a question, for us to achieve 80 marks with 25 questions or with 30 questions, then some of them must be having 2, 3, like that. And therefore, that's paper 1. Come to paper 2. Now in paper 2, we have another 80 marks. But now here, we have only how many questions? There are usually 8 questions. Only 8 questions. Question number 1 to question number 5 is what we call section A. Then question number 6, question number 7 and 8, that is section B. Now in section B, you will answer, section 1 is compulsory. Section A is compulsory, the 5 questions. Section B, question number 6, which is graphical. Now it's what? Compulsory. Then question number 7 and 8, you pick 1. Each one of them comprises how many marks? 20 marks. So we have 20 marks in section B, that is essay, between question number 7 and 8, and then graphical is 20. So section B has 40, section A has 40. That comes to 80. Then we have the paper 3, the practical. Okay? The practical now comprises of how many marks? 40 marks. Now, unless otherwise of the grading system that the government has proposed, unless otherwise, and if there's any change that will come, previously, and many years, it has been that biology paper 3, or any other practical, be it chemistry, paper 3, huwa inakalkuletiwa the way it is. This is what I mean. You get 40 in practical, that is how it is lifted. It's recorded like that, as a percentage, in short. Then paper 1 and paper 2, which a total of 160, shall be now reduced to 60. That is how it has been, unless otherwise. So that's why you keep a lot of emphasis on. If you pass so much in practicals, then the high chance of you performing are very, very high. So, very fast, that's just an introduction. So very fast, because of time, I want us to go to some of the common mistakes. So we shall write some common mistakes. Some common mistakes. And I wish that we go very fast, a little bit. Some common mistakes. And we shall start, and we're going to discuss almost all the topics. So, be ready, okay? So the topic number 1 is classification. Classification. Classification Some of the mistakes, I know I'll be discussing, then what you'll be doing is very simple. Sitaweza dictate all the norms. I'll be discussing, and when I discuss with you, when you see something good that you wish to write, you write. Yes. So you'll be noting what you think you need to know, and what you think that you can just have in your head, go ahead. Yeah. So in classification, classification is the first one, but it is divided into two. We have classification 1, inform 1, and then we have classification 2, inform 2. There's this definition of classification. Classification is simply, it means sorting out or grouping organisms according to their similarity and what we call phylogenetic origin. Sawa? Classifying, sorting, grouping. Sawa? But there is this some words that I'm going to give you. The NEC, the Kenya National Examination Council, currently they accept dicotyledony and monocotyledony as classes. Yes, mnajua. We have classes. They also accept dicotyledony and monocotyledony as classes, but not orders. But angiosperm, that is angiospermatophyta, you know them, eh? Angiospermatophyta and gymnospermatophyta are known as subdivisions. I want to make that very clear. Those are subdivisions. But I want to make it very clear here, because we're talking about some of the common mistakes. They do not accept words like dicot and monocot. Instead of writing monocot, just write monocotyledony. We don't have words like monocot and dicot. Those ones, the neck, when you are doing the marking, zinaonekana kama they are just abbreviations. Kama unataka kuandika dicotyledony na ukaandika dicot. So we don't have, they don't accept that one. When you talk about jointed appendages, there's some people who confuse jointed and joined. The appendages, when you're writing, when you'll be responding to characteristics, tafadhali, say they have jointed appendages, not joined. Are we together? It is not joined, but jointed. Then, be very careful with spellings. Spellings. I'm talking about classification. In spelling now, we have this word arthropoda. Arthropoda. It doesn't have an N. Because some students write what? Anthropoda. We don't have anthro. It is arthro. Arthropoda. With A-R. Arthropoda. Then, when you are using details on characteristics of individuals or individual members of each phylum, that is, or division or classes in arthropoda and chordata, should be avoided. E.g., characteristics of housefly, cockroaches, spiders, etc. Details on characteristics of individual. We don't have characteristics of individuals. Sawa. We have characteristics of either a class, characteristics of a phylum. We don't have characteristics of individuals. Sawa. Yes. Topic number 2, cell physiology. And I'll combine, because of time, I'll combine cell physiology and respiration. Very fast. Again, the cell, we're talking about the cell. We are now in cell physiology and respiration. I want to make it very clear that the cell does not absorb water. The cell does not absorb water. Don't say the cell absorbed water. The cell does not absorb water. What does it do? It draws in. It draws in. It draws in. It does not absorb. It draws. There's a mechanism, and we are talking about the concentration gradient. inapandisha concentration gradient more than the outside, sindio? And therefore by osmosis. By osmosis, not through osmosis. It is by osmosis. So once it has risen, it has risen, the concentration has risen, then it starts drawing in water by osmosis. Don't say absorb through. There's these words. Avoid the words like absorb and through. Another one. The cell, and this one I think of many of the time I always tell you, the cell loses water but does not lose water. There's lose with single O and lose with double O. With single O it means kupoteza. Sidio? With double O means not tight. Not tight. So it is loss, not lose with double O. Tuko pamoja? So when you're saying the cell lose water, avoid using the one with double O. Tunaelewana? The cell sap, or what you call this solution, the solution, tafadhali, I want to make it very clear. The solution of the cell sap, or whatever it is, does not move out of the cell. The solution does not move out of the cell. What moves? What moves? It is water that moves out. The solution does not move. Sawa sawa? And therefore unapatanga umepewa a whisking tubing. Now inside the whisking tubing we have a solution of a starch. You see the solution? Because in starch plus what? Sindio? Then umekwambia umechukua hiyo umeimass ama tuseme it's a solution of sugar, whatever it is. Then umeweka ndani ya sugar, iko na sugar na umeweka ndani ya maji. So definitely we will start drawing in water. I want to say it very clear here. Don't say the solution gained. Don't say the solution lost. What is making it to gain or lose is what? Water. It is the water that imefanya ukaona hii imepungua na hii imefanya nini? So talk about the water, but not solution. The solution does not move, but it is the water in the solution that does what? Move. Because a solution is a combination of a solute and a solvent. So it is the solvent that is moving out, leaving the solute. So the solution does not move, but water moves. There's these words that I want to make it very clear. There's these words: plasmolysis, flaccid, and turgid. The three words: plasmolysis, flaccid, and turgid. Plasmolysis, the three words I'm mentioning them, plasmolysis, flaccid, and turgid. Those ones are words which are used when we are talking about which kind of a cell? A plant cell. Don't say an animal cell was flaccid, or was plasmolized. No. We are talking about, anytime we are talking about a plant cell, that is when you mention flaccid, a plant cell, when you mention flaccid, plasmolysis, and turgid. But when you're talking about an animal cell, there are these words that you use. You use words like crinated, studio. Yes, they are ringled, hymolized. Those are the words. Hymolized. Another thing, writing of abbreviations should be avoided. Like you want to write the word endoplasmic reticulum. Then you remember that the spelling maybe is Takusumbua, then you write ER. Or RER, rough endoplasmic reticulum. No. Abbreviations, and I'm repeating, abbreviations are not accepted. Sawa sawa. And I've given you examples. When you're talking about the microscope, it has what we call a revolving nose. A revolving nose, some people are talking about the revolving nose hold. They hold the objective lenses. Holding is this, you hold. But the work of the revolving nose is to do what? To support. not to hold. Unajua ku hold ni kushikilia. Yaani kama inafaa tu ishikilie na iachilie, hapana. It is supporting. Sawa sawa? So don't say the revolving nose holds. What does it do? Supports the objective lenses. The function of mitochondria. I know this is where we confuse much. The function of mitochondria. I want somebody to tell me, what is the function of mitochondria? What is the function of mitochondria? Yes, it is site for respiration and not for respiration. Some people say mitochondria is for respiration. Avoid such kind of word. Another word, another organelle is ribosomes. Ribosomes, we don't say synthesizes, but what does it do? It gives sites for protein synthesis. It gives sites for protein synthesis. Inakupatia nafasi ufanye vitu zako. Give you what? Sites for protein synthesis. But does not what? It does not synthesize. Some people say synthesizes protein. No, it doesn't synthesize. It only gives site for protein synthesis. Sawa sawa? It only gives site. Another one, respiration. After I said I'll combine the two because of the cells. Respiration does not only take place in mitochondria. Because of what? Because of what? Because of glycolysis. Where does glycolysis take place? In the cytoplasm. Studio? Glycolysis takes place in the cytoplasm. Is cytoplasm inside the mitochondria? No, it's not. So that means respiration does not necessarily take place alone inside the mitochondria, but it also takes place outside. Now, respiration is different from gaseous exchange or breathing. Tunaelewana? Because of this thing called respiratory surface, some people start confusing between the respiration and gaseous exchange. When you're giving the definition for respiration for gaseous exchange, we mentioned something to do with what? Respiratory surface. So some people start confusing that maybe respiration has a relationship with gaseous exchange. They are not. Now, when you are using the terms, there are these terms that we use: hypertonic, isotonic, and hypotonic. You must use them comparatively. Comparatively. What does it mean? You must compare. For example, the cell is hypertonic to. Comparatively. You cannot say it's isotonic to what? So, when I go down there, you must compare something to. When you're talking about the solution is hypertonic. Hypertonic to what? It's hypotonic. Hypotonic to what? So the cell sap is hypertonic to the solution. So?
Yes.
You are trying to give the reason why whatever is happening is happening. Now, isotonic is not the same as neutral. Isotonic is not the same as neutral. Why am I saying that? I may be having a cell which has a certain concentration, which is say 2% of whatever, 2% of a certain solvent. Then you also have the same solution which has 2%. When you bring those solutions together, we shall say they are isotonic. But are they neutral? They are not. This one has solvents, this one has solvents. They have the same concentration. So isotonic does not mean neutral. And I know you understand this, no?
Yes.
It does not mean neutral. Another thing, we talk about lysosomes. What is the function of lysosomes? They destroy worn-outs. But we don't say. There's some people who say it destroys non-functional. No. Non-functional. And it destroys non-functional. Be very clear. It destroys. But does it destroy? First of all, I'm saying it destroys. Does it destroy? It doesn't destroy. Correct. Lysosomes does not destroy. What it does, it contains lytic enzymes, which does what? Are we together? Yes. It contains. So when you're giving your answer, just say lysosome contains lytic enzymes which destroys. So themselves they don't destroy, but what destroys is what they carry, is what destroys. Are we together? Yes. Yes. Very fast. Nutrition. Nutrition. So for more information you can consult, isn't it? Nutrition. I'll be very fast. Number one is teeth. Teeth are not for chewing.
How?
Teeth are not for chewing. When I say teeth, I mean all of them. We have different types of teeth. We have incisors, canines, premolars, molars. Look over one. What is the function of incisors?
Cutting.
What is the function of premolars? So when you say teeth are for chewing, what are you saying? You are saying that I have a homodont, eh? Homodont, all of them are just chew. I don't cut. My Kcse is to chew. So teeth are not for chewing. I.e., if you are writing, incisors are for cutting, canines are for ripping off, molars and premolars are for grinding. For those who are writing. Another thing, the jawbone. The jawbone does not hold teeth. Don't say the jawbone holds teeth. What does it do? It supports teeth. Supports? It supports or secure them firmly. Don't say that the teeth are sharp. Teeth are not sharp. What is sharp? Be very specific. Incisors, they have a wedge shape. They have a wedge shape for cutting surfaces. Canines have pointed cutting surfaces. Premolars have flattened, not flat, broad. Some people say flat and broad. They are flattened, not flat, broad. Are we together? Yes. Not flat and broad. They are flattened. Enamel, not enamel, the outer layer of the teeth. Enamel protects the tooth from mechanical damage, not damage. It does not protect it from being damaged, but it protects it from what you call mechanical damage. Let's switch off the I've talked about enamel protects the teeth from mechanical damage and not damage. Another thing, chlorophyll. Chlorophyll doesn't absorb light. It absorbs light. What does it do? It traps light. It doesn't absorb. It traps light. The lamina or the leaf is not flat, but is what? We talk about the leaf. The leaf is not flat, but is what? Flattened. It is flattened. It's not flat. It is flattened. The leaf apex is not sharp. The leaf apex, it is not sharp. It is what? Pointed. It is not sharp. So they don't say that the leaf apex is sharp. No, the leaf apex is pointed. Thick leaves, or what you call the thick leaves, are not fresh, but they are fleshy. I know people will say, they are not fresh, they are fleshy. Fleshy. And remember the word fresh and fleshy. If you change L and R kidogo, fresh inamaanisha very very new, sindio? But fleshy inamaanisha it is succulent, iko na uzito fulani, juicy. The cuticle protects a leaf against what we call mechanical damage. The leaf protects the cuticle protects the leaf against mechanical damage. Let me go very fast a little bit. Essential amino acids. We talk about, there are two types of amino acids. Essential and non-essential. Essential amino acids are those that cannot be synthesized by the animal's body, not body. By the animal's body, not body. When you just say body, which one do you mean? You must be very specific. They cannot be synthesized by the animal's body, but not body alone. Tuko pamoja?
Yes.
Enzymes do not die. And enzymes cannot die. But they are what? But they are what?
Denatured.
They are denatured. Enzymes are denatured. Saprophytes, you know saprophytes, what does it mean? They feed on what? They feed on dead, decaying matter and not on hosts. So be very careful when it comes to that. Transport, let's go to another topic. Transport in plants and animals. Transport in plants and animals. Let us talk about the capillaries. The first thing we talk about is capillaries. Because when you're talking about transport, we talk about the arteries, the veins, and the capillaries. You know, when we are giving the characteristics of the arteries, what do we say? We say that the capillaries are one cell thick. But don't say that the capillaries are thin. Oh, they are not thin. It is their size that they are one cell thick. One cell thick. But don't say they are thin. They are not thin. They are one cell thick. Blood vessels do not contract and relax. Blood vessels do not contract and relax. What contracts and relaxes? Muscles. So it is the muscles. It is the muscles they have that do so. So what do I mean? This is what I mean. Contraction and relaxation. Anytime you are mentioning contraction and relaxation, should be related to muscles. It's only the muscles that can contract and relax. So if you are seeing anything contracting and relaxing, what does it have? Muscles. So it is the muscles inside that whatever it is contracting that is causing those contractions. Don't say now it is the heart contracting. No, it is the muscles at that heart that is doing what? Is contracting. Another thing is cross sections have sections of plant stems. Cross sections have sections of plant stems. Roots should have double outline when you are drawing. Roots should have double outline. Then gaseous exchange in plants and animals. Gaseous exchange. We are now in form. We are now in form. In form 2. Gaseous exchange in plants and animals. I'll start by looking at the rib. When you are explaining the breathing in and breathing out, breathing in and breathing out, I want to make it very clear. The rib cage doesn't contract and relax. The rib cage does not contract or relax. What contracts and relaxes? I've already mentioned. The what? Muscles. And for this case, we have what we call the intercostal muscles. So we have both external and internal. And we know when it comes to muscles they work in the opposite. I don't know which term to use. When one contracts, the other one relaxes. And when the other one relaxes, the other one contracts. So, So when you are explaining, when the ribs are moving outside, remember it is the external intercostal muscle that does what? Contracts. And the internal intercostal muscle does what? Relax. The gills, the lungs, they are highly vascularized. Why are they highly vascularized? Highly vascularized, the they have large number of blood capillaries. Why are they having large number of blood capillaries? The reason is to do what? To facilitate transportation of gases. To do what? To facilitate the transportation of gases. Then another thing, why are they moist? I want you to be very careful as I give you. You are able to facilitate transportation of gases. Then why are they moist? To do what? To dissolve gases. To dissolve gases. We have gill filaments. You know the gill filaments, eh? What is the function of gill filaments? Gill filaments? It is to do what? To increase the surface area for maximum diffusion of gases. There is this, I want to explain to you that the starch formed in the gut cell, you know there's a starch formed in the gut cell, yeah? It's not osmotically active, but lowers the gradient of the cells. It is not osmotically active, but lowers the osmotic gradient of the cells. Starch formed in the gut cell is not osmotically active, but lowers the osmotic gradient of the cell. Then when we are testing for presence of, we are still talking about Kcse, when we are testing the presence of carbon 4 oxide, what do we use? If you want to know that there's carbon 4 oxide in the carbon 4 oxide is coming out as a byproduct in any reaction, what do you use? You use lime water. The name for lime water is calcium hydroxide. It's calcium hydroxide. Yeah? So when you're using that lime water, when you're using the lime water, and you pass the gas, you know, when you pass the gas through the lime water, what happens? It forms white precipitate, and we have said we write white precipitate, not white PPT. It forms white precipitate. Tumesema abbreviations are not used, you know? Yes. White precipitate, not milky. It forms milky, you know, it forms a substance which is white, it forms jewel, it forms a colloquial. My friend, it forms a white precipitate. Gut cells do not close and open stomatas. What do they do? They control or regulate. Don't say they open and close. And the gut says nafungua na kufunga. They control or do what? Regulate. So the other one. Yes. Excretion and homeostasis. Excretion and homeostasis. Now, tukianzia to the word excretion. There are some people who confuse the spelling of excretion. It is excretion. There is no A there. Between E and T, there is no A. I'm sure even some of you have written with an A there. Excretion. No, excretion should not be confused with excretion. Excretion should not be confused with excretion. There's a difference between excreting and excreting. So excretion should not be confused with excretion. Likewise, secretion should not be confused with secretion. Secretion should not be confused with what? Secretion. Next, there's this common term, germination. What is germination? I know it's the function of the liver. One of the functions of the liver is what? It performs germination. What do you understand by germination? Yes? Okay, let me give you I want to give you because and then you tell me if it is true or. Germination is the breakdown of amino acids by the liver cells. Germination is the breakdown of excess amino acids by the liver cells. Which one is true? I've given you two definitions. Excess. You must mention the word excess. Germination haiko hapa kuvunja tu any amino acid naona. Inavunja ile yenye iko excess. So when you ask define the word germination, make sure you mention the word excess. It is the excess part that will be germinated. Sawa? It's the excess part that will be removed. But the normal one shall be consumed by the body. It's excess that will be eliminated. There's also insulin and glucagon hormones. And I want to say here very clearly, insulin and glucagon hormones do not regulate amount of blood sugar in the body. They do not. Sawa. That is not their function. They do know that. They are fanyangiyo. But what do? What is their function? They stimulate. They do. They stimulate the liver cells to regulate the blood sugar. They stimulate. So there's transition. It's time now to break down. It's time to. They stimulate. Another one, blood enters the glomeruli before entering the nephron. Blood enters the glomeruli before entering the nephron. Is it true? Where does the nephron start from? Why? Why am I saying that one? It's because glomerulus is not part of the nephron. It is not. It is not. Now you know. It is not. It is because it is not. Now you know. Yeah, I know. Now you know. Isn't it? So it is not. Let us move to ecology. Number one, in ecology we have primary producers. Primary producers. You have producers and you have primary producers. And then there's this word adapt and adopt. Adapt and adopt. There should be a very clear distinction between those two words. To adapt and to adopt. To adopt ni kuchukulia, sio? Kuadapt ni kukubali, sio? When the environment is like that, you adapt to it, sio? You do what it wants, eh? But to adopt, you pick and use it, sio? Misspellings. These are spellings. The word that is commonly misspelled. It's good that I tell you. The word that is commonly misspelled is the word rhizobium. Rhizobium. What are rhizobiums? Rhizobium are bacteria found in the root what? Nodules of leguminous plants. So rhizobium is some students they confuse with what? Rhizobium. There's an M they put there. Rhizobium. It is rhizobium. And then we are talking about nitrogen fixation. Which part of the plant does nitrogen fixation? It's supposed to be nitrogen fixation in the soil, not root nodules in case of leguminous plants. Can I take it again? You should talk of nitrogen fixation in the soil, but not root nodules. Not in the root nodules, but in the soil. Nitrogen fixation is done where? In the soil, but not in the root. Tuko pamoja?
Yes.
I want to see if there's a difference, and I want you to help me. Is there a difference between submerged and floating plants? Submerged and floating. There are floating plants, and those are which are submerged in those things. Hence, I say submerged, half of it is in water, half of it is on. Then floating, the entire of it is on. Anyway, the only thing I want to tell you is submerged and floating are different. They are not the same. So, yes, the way he's putting it. Submerged and floating are not the same. So, ukifikia mahali tunafaa kutumia submerged, say they are submerged. Looking at maybe the structure the way it is submerged, the way you say, or it is floating. But it is not the same. It does not mean the same thing. Reproduction. Reproduction in plants and animals. Tuanze na petals, sepals, na stamens. What nasema they are? They are fused. They are? But don't say they are adjoined. They are fused. It's not fused. They are not joined. Petals, it is not the flower that attracts insects. It is the petals. And how do the petals look like? They are brightly colored to attract what? They don't attract insects. What about if something else comes and it's not an insect? We shall say, no, don't come here, I will not attract you. It attracts pollinators, whatever they are. So it is brightly colored to attract what? Pollinators. Whatever mwenye atakuja. Si hata wewe unaweza kuwa pollinator. Some of you look at a flower and you say, wow, that flower is nice. So it attracts you, you go and pick it. As you pick it, you shake the pollen. Inamwagikia ingine, so you are an insect. You are not an insect. You are just a pollinator. Who was attracted by what? By the petals. Seeds or fruits dispersed by self-explosive mechanisms and not self-dispersal. Which mechanism? They are dispersed by self-explosive mechanism and not self-dispersal. When you are giving you a distinction, distinguish between fruits, seeds, flowers, and nectar. When you are giving a distinction, make sure fruits, seeds, flowers, and nectar, they are different. Now, there's this one that I want us to look at, placenta. Placenta is a medium of exchange of material between mother and fetus. Let me read again. Placenta is a medium of exchange of material between mother and fetus. And I want to be very clear here. It is the fetus, not the baby, not the child. It is the what? Fetus. Don't say it is the exchange between the mother and the baby. It is the fetus. While the amniotic fluid, is this amniotic fluid? Amniotic fluid does what? It does not prevent. What does it do?
Absorb.
It absorbs the shock. Absorbs shock. Progesterone hormone. Progesterone hormone does not prepare mammalian female uterus for pregnancy. What does it do? Progesterone hormone does not prepare prepare the mammalian female uterus for pregnancy. But the bursting open of the Graafian follicle to release what we call immature ovum. But what anapeana function ya progesterone? It helps in the bursting or opening of the Graafian follicle to release the immature ovum. Growth and development. Growth and development. Sasa na hii hormone ambayo wewe inasumbua watu sana inaitwa ecdysone. Ecdysone, sio? Ecdysone hormone inafanyanga nini? Ecdysone hormone inafanyanga nini? Ecdysone hormone inafanyanga nini? It's a multi hormone. But what does it do? It controls the multi. Sawa. It controls. So unaandika unasema ecdysone hormone controls multi, but not growth. It does not control growth. Now, remember what tunasema hivi. Growth is an increase in what? It's a permanent increase in size. Now, when an organism, for example, this one we're talking about the arthropods, when they shed the exoskeleton, that is molting, they increase. They increase. Yes? It is the exoskeleton is increased, but growth has been taking place inside. Tunaelewana? Yes. So hii hormone haikuji kufanya grow, growing has been taking place. What is inakuja kufanya hii hormone ni kuja tu kushed so that it can increase inside. Tunaelewana? Yes. So it does not help in growing, but it helps in what? Molting. Tunaelewana? Then we have epigeal germination, or what you call the epigene. You can pronounce the way maybe we'll do that. Epigeal or epigene. So in epigeal germination, the seed coat and cotyledon emerges out of the ground. But don't say the seed comes out. That's a seed kitoka nje na imesha germinate. There's some people who say that in epigene the seed comes out. Na ikitoka nje imegerminate. It is the cotyledons after germination. What remains? The cotyledon which come. Yes. And I want you to be very specific when answering such kind of question. The seed coat is made of two levels. The outer layer known as the testa and the inner layer known as what? The tegmen. Maize. What is maize? Maize is a caryopsis and not a seed. Maize is a caryopsis and not a seed. Why? Why do you think it's a caryopsis and not a seed? Because it has fused seed coat and pericarp. It has a fused seed coat and pericarp. What is a seed dormancy? That's a period between. That's a period that a seed undergoes after ripening and before germination. So it is a period. It's a period that a seed undergoes after ripening and before germinating. Now let us go to genetics. I said I'll be very fast because of time, so that we can cover as much as possible. For more information, we shall be consulting and we shall be talking. So genetics. Number one, what is so important in genetics? When you are doing your process, I want to be very specific. When you are doing your process, tafadhali, let us use letter symbols which have respect or with respect to the genes we are talking about. If you are talking about red color and use letter V, are you being sincere to yourself? If you are talking about the red color, use which letter? Use R. Sawa sawa. So let the letter represent the dominant gene in terms of its capital letter represent the dominant, and then the small letter to represent the what? The recessive gene. And then these letters that you write, they must be cycled. They must be? They must be circled. Is it circle or cycle? Circle. It must be circled. But when you are doing your process, when you write across between the when you are doing kuna phenotypes ndio, kuna genotypes na kuna gametes ndio. So pale kwa genotypes hapo kwa parental genotype hapo ndo tunaweka nini? Unaelewa? Away kwa phenotype tafadhali unaweka kwa genotypes. That's where it is the genes that cross, the genotypes that cross. There's not much about that one. We can move to reception, response, and coordination. Reception, response, and coordination. And I want to give you some words here that I know you maybe you have met them. Short-sightedness does not mean short-sighted. Yeah? Short-sightedness does not mean short-sightedness does not mean what? Does not mean short-sighted. But it's a what? It's a condition in which rays from a distant object converge in front of the retina. Rays from a distant object, they converge before reaching the retina. And if they converge before reaching the retina, what will happen? There will be no image. So these individuals wanaona blood images from a distance, wanaona zikiwa blood because they are formed before reaching the retina. But it's very wrong to say that the image from a distant object is brought to focus in front of the retina. Are you seeing the difference? That the image from a distant object is brought to focus in front of the retina. If it is brought to focus, then it's supposed to be perceived, see? Yes. Yeah. So it is supposed to be rays from a distant object converges in front of the retina. Converges, but they don't bring into focus. They converge before reaching the retina. What about long-sightedness? Long-sightedness also does not mean long sight. Haimaanisha naona mbali. But it's a condition in which rays from a near object converges beyond the retina. But it's also wrong to say that the image from a near object is brought to focus behind the retina. But they converge beyond the retina. The ear pinna, this one, what does it do? What I'm giving you is common mistakes that we do. The ear pinna, what does it do?
It traps sound.
It traps
Sound waves.
It traps sound waves. Yes, so I want to say it does not trap. It concentrates. It concentrates. So concentrates sound waves into the ear canals, but it does not trap. Don't say it traps. Could trap ni kushika. That is trapping. But this one, it concentrates them. Yaani unazikusanya unaziweka mahali pamoja. Inatoka huku naikusanya na unaingiza kwa hiyo unakusanya, una concentrate, si ndio? But how trap ni maanisha unashika na haupeani, si ndio? Yes. Unaelewana? Then when answering questions on adaptation of the eye, to its function, you must, accessory structures should not be mentioned. When you talk about the accessory structures, should not be mentioned. Accessory structures should not be mentioned. Tunaelewana? Aya, very fast. I think we are almost towards the end. I want to talk about some of the languages used and how you respond. Languages, just write languages used in biology examination. Languages used in biology examination. Languages used. And here I'm talking about, I'm going to mention there are around 18. I'll be very fast. There are around 18 terms. I think you'll give me, you'll add me some 30 minutes. Yes. Some 30 minutes. So in the 30 minutes time so that I can complete completely. Yes. I'm saying, or you want us to have a break?
No.
Then we can continue. So I was saying, languages used in biology. There are these languages, because some of us, and I'm sure some of you were here, some of the students, they fail biology because me I know. Some of the students they fail biology, not because they don't know, but it's because they are responding a wrong things. Whatever they have been asked and the answers they are giving, they have no relationship. So I want to give you some of the terms. Term number one that we use commonly in biology is with reference to. And I'll give you a question. Hapa nitakuwa nasoma nikipeana Kiswahili, si ndio? Napeana na jibu. Sio ni mzuri? Yaani wewe utakuwa na kazi leo. Mimi napeana swali, napeana mpaka jibu. Lakini venye nitakuuliza pia utajibu, si ndio? Yes. Swali number one is with reference to. What does it mean? Before I give you an example, what does it mean with reference to? It means you must use specific examples to make the meaning clear. You must use specific examples. That's why they're saying with reference to. So you must use specific examples. Now, example, with reference to the leaf structure only. With reference to the leaf structure only. Explain how xerophytes are adapted to their habitat. With reference to the leaf structure, explain how xerophytes are adapted to their habitat. So what does it mean? This is what it means. Maybe I can just be giving you what you are not supposed to do. So, this is what it means. If you state the features, if you do what? If you state the features of roots and stems in the question, you won't get the marks. Because they are very specific. What have they said? With reference to what? With reference to the leaf structure. So if you start talking about the roots, it has dark roots, it has you have missed the point. We are only talking about the leaf structure. Number two, I said let me just be giving you what you're not supposed to do. The answers are so many if I dictate. Number two, list, name, and give. List, name, and give. List, name, and give. Wanakwambianga list 2, name 5, give 4. Sawa sawa? Yes. So what are you supposed to do if you are asked to list, name, and give? Here, you are supposed to write down the points one by one without explaining them. You are supposed to write the points one by one without explaining. You are only supposed to list, name, or give. So you are supposed to give the point one by one without explaining. For example, name the process by which mineral salts enter into the plant. Name the process by which mineral salts enter into the plant. Can you give the answer? Active? Active? Transport. Another one? And I can give you another question. So, name the process by which human body naturally stops bleeding. Name the process by which that is blood clotting. Correct. Number 3 is suggest. Number 3 is suggest. Suggest. Unapata swali inasema suggest. What are you supposed to do with that kind of a question? This one means giving possible reasons for something. Giving possible reasons for something. An example is here. Example, inasema hivi. This is an example. Suggest an explanation for increased number of red blood cells found in blood of people normally living at high altitudes. Suggest an explanation for increased number of red blood cells found in the blood of people normally living at high altitudes. So, suggest. Mbona hawa watu wenye wanapatikana kwa high altitude have higher number or large number of red blood cells in their blood? To transport? But they are asking you to suggest an explanation for the increased. So you must first of all explain why they are having that look. Now they are having the high number of red blood cells. Why? Why? This is because, Madam is saying, this is because at high altitudes, there is what you call low air pressure. Ukipanda juu, unakuwa na low air pressure. And low air pressure huwa inafanya nini? Huwa ina reduce the concentration of oxygen. So hence, low oxygen concentration. And therefore, the body does what? The body responds. Or sometimes they say it acclimatizes or acclimates. Acclimates by doing what? By coming up with more red blood cells in order to carry enough oxygen for transportation. Number 4, trace. Trace. Trace here. T-R-A-C-E. Trace. Si mnajua ku trace? We all hata primary zimekuwa mkitrace. So what does tracing mean? It means to follow the track of something. To follow the track of something in drawing. To follow the track of something in drawing. So what does it mean when you are responding to questions? It simply means to make an exact copy of. Make an exact copy of. Kufuata tu. To make an exact copy. Like if you are tracing the map of Kenya. I know when we used to be in primary, we used to do that, because you don't know how to draw. Your hands are not yet established. Your muscles are not strong. So wakwambia chora map of Africa, you take the textbook, nawekelea hapa with a pencil, then you trace, so that what you get at the end is an exact copy. So what are you doing? You're only coming up with an exact copy. So tunasema you are tracing. An example: trace the path taken by molecules of oxygen from the atmosphere to the respiratory liver tissue in man. Trace the path taken by a molecule of oxygen from the atmosphere to the respiratory. That is the respiratory. Trace the path taken by a molecule of oxygen from the atmosphere to the respiring liver tissue in man. So, if that is the case, then what are you supposed to respond to this question? How are you supposed to, I will explain how you're going to respond to that question. How do you respond to this question? Because it is asking you to trace, then it means you will have to explain. You have to explain first how oxygen enters the body. So, because you are tracing, you give an exact copy. How oxygen enters the body. Then how it enters the blood vessels. Then how it enters the organs. And then you state the changes that occur to the oxygen as it's moving to the liver tissues. In other words, you give the route taken. So you have to follow it to the end. From the entrance to the exit. Number 5. To what extent? To what extent? To what extent? What does it mean? This means what effect something has. What effect something has. What effect something has. Example, to what extent does the hormone insulin affect sugar level in the blood? To what extent does the hormone insulin affect sugar level in the blood? To what extent? Okay? So when you're answering this question, this one I can give you the answer because it's short. Insulin increases oxidative breakdown of glucose. It increases oxidative breakdown of glucose and facilitates the conversion of glucose to glycogen and fat. Glycogen and fat. Number 6 is with illustrations. With illustrations. With illustrations. So this one means what? This means to explain something using specific examples. Explain something using specific examples. Example, I'll give you an example. With illustrations, describe how new plants arise by asexual reproduction. With illustrations, describe how new plants arise by asexual reproduction. So, utaongelea kuhusu vitu kama binary fission na una explain, by fragmentation na pia una explain, by sporulation, sindio? Na pia una explain. That is with illustrations. Adapted to. Hapa ndio wanafunzi wengi wanashindwa sana, sio? Adapted to. Hapa ndio wanafunzi wanashindwa sana. A question. Describe how the red blood cells are adapted to their functions. Describe how red blood cells are adapted to their function. So I want you to note this one. You have no note. Note. N-O-T-E, capital. You know this one. Anytime you ask a question on adaptation, the function is tied to the feature. The function is tied to the feature. And therefore, the two must be mentioned. The two must be mentioned. The feature it has and the function zinakuwa zimeshikana. Tunaelewana? Adaptation of red blood cell to its function. You must, wakati tunasema it lacks a nucleus. Sio? Yes. Wakati tunasema the nucleus is absent. Then why? The question that follows is why? The feature should be tied to what? The function. Zimefungwa pamoja. Tunaelewana? When you say it is by concave in shape, by concave in shape, why? Sawa sawa? Are we together? So the feature is tied to the function. Another number 8 is discuss. Discuss. And the question for example, the example I'm giving you is discuss the various evidences of organic evolution. Discuss the various evidences of organic. Give an example. Discuss the various evidences of organic evolution. What does discuss mean? Discuss is open. This one means it means to examine something critically and to exchange ideas and views. Can you discuss? That is to examine critically and give what? Give your ideas and views. That is now a discussion. Okay? So we discuss critically by explaining. And therefore, in discussing once can be for or against the idea or both. In discussing, one can be for or against the idea or both. What do I mean? Mtu anaweza support ama akatae. So unaona kwa darasa mnatoa maoni, si ndio? Alafu mwingine anasema mimi naona kama tusifanye hivyo. Si ndio? Mimi naonelea tufanye hivi. Na kuna wengine wanainama wanasema tunamuunga mkono, si ndio? So in discussing, one can be for it, for not, or for both. Tumeelewana? Account for. Another problem. Where another problem? Account for. So I know when it comes to account for, that's where the biggest problem lies. Account for. Account for. Definitely, definitely, I'm sure each one of these are affected with accounting. Yes. The most affected are the business students. If they see a question there inasema account for, wanakumbana to accounting. Unajua hapa ni hesabu ya accounting. We are accounting for. So when you're accounting for, it means, in biology, accounting for means to give reasons as to why something happens or happened. To give reason as to why something happens or happened. Sasa unapeana reason. For example, question number 6, wewe inakuja sana na hiyo maswali ya account for the shape of the graph between point A and B. So the first thing, the most of those questions zinakuanga 3 marks ama 2 marks. Haiwezi kuwa 1. Kwa sababu one of the first mark is lazima kwanza useme chenye kina happen. Sawa? The rate of reaction is increasing with time. The rate of reaction is decreasing with time. Whatever you are seeing with your eyes, you get a mark for that. The level of progesterone is increasing with time. Or the level of progesterone is reducing with increase in estrogen. When estrogen increasing, the level of progesterone is decreasing. Kusema tu hiyo peke yake, you get a mark. Then you explain why. Why it's happening. Then you get the other mark. Sawa. So anytime you get a question and saying accounting, first of all state what is happening, then explain. What is happening, then explain. Kama utakosa kujua explain, utapata mark ya what is happening. Because what is happening you can see with your eyes. Ndio? One is increasing, one is decreasing. So kama ni 2 marks uko na moja. You will never fail kujua kwa nini inafanyika, but you already have a mark. Describe. Now, to describe means to present the features of something in writing. To present the features of something in writing. To present the features of something in writing. For example, describe how hormones regulate the menstrual cycle in human beings. Describe how hormones regulate menstrual cycle in human beings. So here you have to name the hormones. Sawa. Because they are saying describe how hormones. Don't just say those hormones they do this. Those hormones they do this. And you see the hormones, yeah, they also do this. No, you must name them. Estrogen does this and this. Progesterone does this and this. Luteinizing hormone, hakuna hormone inaitwa LH. Luteinizing hormone. Sawa. Hakuna hormone inaitwa FSH. At follicle stimulating hormone, sasa una haraka. Ni FSH. My friend. Hiyo mimi nikimark na wekanga tu na ku underline. Hiyo mimi sijui ni FSH ni nini sasa? FSH inaweza kwa nini? Inaweza kwa kitu yote. Full scholarship home, ndio. Full scholarship. It can be something else. So wewe niandikia follicle stimulating hata unaandikia LH. LH ni nini? So write all those, and then any hormone you write, you do what? You give an explanation to it. But you can write follicle-stimulating hormone in brackets, FSH, that's your choice. But if you write FSH alone, you go home dry. Give another one. Give a description of. Give a description of. Give a description of. Now, in giving a description of, this means, so I hope you are right, this means This means to present the features of something in writing or speech. This means to present the features of something in writing or speech. Example. Describe how hormones regulate. Oh, yeah, nimekupatia. Describe. Oh, give description of. We are on give description of. Oh, sorry. This means to make plain or to make it clear. To make plain or to make it clear. To make plain or make it clear. Example. Explain the role of light in photosynthesis. Explain the role of light in photosynthesis. Explain the role of light in photosynthesis. So, when you are giving your answer, you do what? You talk about the light provides what? Energy. Light provides energy. Number 12 is what is significance of? What is the significance of? What is the significance of? What is the significance of? This means the importance of. The importance of something. The importance of something. For example, what is the significance of crossing over? What is the significance of crossing over that occur during the process 1 of meiotic division? What is the significance of crossing over that occurs in process 1 of meiotic division? That's an example that you can look at. Number 13, define. Define. This means to state precisely what is meant by or what is comprised in something. This means to state precisely what is meant by or what is comprised in something. What is meant by or what is comprised in something. For example, define osmosis, define diffusion, define photosynthesis. What are we giving? We are doing what? We are stating precisely what is meant by or what is comprised in something. State. Inamaanisha nini? Presenting your answer in statement form. Presenting your responses or answer in statement form. For example, state the role of water in seed germination. State the role of water in seed germination. Another one. What is meant by. What is meant by? What is meant by? So here you give the meaning of something using examples where possible. Give the meaning of something using examples where possible. Example, what is meant by the term speciation? What is meant by the term speciation? I'm trying to finish so that we can wind up. Number 16, distinguish or differentiate. Distinguish or differentiate. This means to bring out the differences. It means to bring out the differences. This means to bring out the differences. But I want to say something here. Please, when you are giving the differences, there's a difference between differentiating and distinguishing. If you are asked to distinguish and you give one, for example, distinguish between osmosis and diffusion. Now unajua tu osmosis, haujui diffusion. Kama umekuuliza kudistinguish, utapata mark ya osmosis. Lakini kama umekuuliza kudifferentiate na haujui moja, my friend, you are wasting time. Kama haujui moja, hiyo ni ink una waste. Kwa sababu hauja differentiate. But kudistinguish ni kama kutoanisha pamoja, sio? So umetoana moja umesema hii, haujui hiyo nyingine lakini umesema hii. So uko sawa. But to differentiate you must state what this is and this one is. Sawa sawa? How this one looks like and this one looks like. And don't say this one is hot and this one is tall. Abana. If this one is hot, this one is cold. This one is tall, this one is short. Don't say this one is tall and this one is taller. Don't have tall and taller. You say that you are tall and short. And this one is short and this one is a dwarf. What is the difference there? That's not a difference. You must give a distinctive word, characteristic. Give reasons why. Give reasons why. So this means you have to give reasons why something is so. Give reasons why. So you have to give reasons why something is so. For example, they are saying, give reasons why it's only mutation in genes of gametes that influence evolution. Give reason why it's only mutation in genes of gametes that influence evolution. The last one is, with the help of a diagram. With the help of a diagram. Yours is self-explanatory, isn't it? Yes. That means you must draw a well-labeled diagram to assist you explain the points. You must draw a well-labeled diagram to assist you explain a point. Now, just to wind up, take a please. There are so much that we can discuss about biology and we cannot finish today. So this is just a brief so that you, as from today, you start now reconstructing your mind. You start now rearranging yourself. There's some words I've given you where you think that I'm not clear. You ask again. Mwalimu haya, you say that I do this and this one. What about if I meet a question which looks like this? The only thing I can do is I'll advise you where I can. But now from today, this should be like your guidelines, so that you know this is what I'm supposed to do and this one is not what I'm supposed to Sawa sawa? Because at the end of the day, once you sit for that paper, the marking shall be standardized. Marking equal the same countrywide. If we agree that we're going to mark spellings, mbona sijataja neno to do with spellings? Because nowadays huwa ina fluctuate. It does not, it is not fixed. Sometimes wanakwambia forget about the spelling, sometimes wanakwambia look at those spellings. Sawa sawa? So we are not sure of that, but there are some ones which nimekupatia pale, which you must be very careful of. Because English, when it comes to the English, the way you present your content, English matters a lot. You can present your point, somebody misunderstands those points. Why? Because the way you are presented is not the way you are thinking. I told you about the English that we think and the one that we speak. Are they the same? They are not. The way you present and the way you wanted them to come out, it is not the same. But through practice, daily practice, and with the reference of those notes, you will now start improving slowly by slowly. So that by the time we sit for that exam, by the time you enter that room of the exam, you are well equipped. But also, don't keep these things so much in your head. This is just a reference. Especially kila wakati ukisoma swali unakimbia kwenda kwa na mwalimu alisema nini? Hautawahi move. Sio? Hii soma, pitia, then do your work. Do your work. Sawa sawa? Any question? Because I can see it's lunchtime and I know what lunch means and today is Wednesday. I know what I've done to you. Some of you are under distraction. Roho zimekufa. They are not sure if they will get something up there. I can assure you will get. Yes. Otherwise, thank you very much for listening. Thank you very much for listening. And once again, I think I've finished. I'll cut at that point, so don't worry about the recording. Please, for those ones who go to YouTube and they watch, kienda pale, look for my name, you'll find me. Now, please revise with me. As today I'm also, kuna karatasi nataka kuweka this afternoon. Weka enda pale, sikiza mambo mawili matatu kapa. Wakati umelala umetulia pale, wekelea YouTube, weka. Wacha kukaa tu naenda sijui unafanya nini. Una watch wa mlambezi wa mnyonyewe. Weka hii. Hii mambo ya Kcse, wacha iende kando bwana. Saa hii sasa unacheza na utafuta mwalimu wa chemistry online, utafuta mwalimu wa CRE online, sio? Kcse ni wakati gani? Wakati umemaliza tu December. Wekelea Kcse, sio? Hiyo ni sawa. Otherwise, thank you very much and madam, back to you.